Welcome to Quick MCAT Lessons, where I try to explain MCAT topics in 10 minutes or less. Today we'll talk about retinal cell types, signal flow, feature detection, and parallel processing. So first let's go over some terms regarding visual sensory information. Transmission. This is the electrical activation of one neuron by another neuron. Perception is the conscious sensory experience of neural processing. Processing is the neural transformation of multiple neural signals into a perception. Transduction is where energy is transformed from one form to another. So this could be light energy to electrical energy by rods and cones. Sensation requires a physical stimulus to be converted into a neural impulse, and light is converted to a neural impulse by a photoreceptor. The light travels in a counterintuitive way. It enters the eye and it passes through the ganglion, horizontal, amacrine, and bipolar cells, and it reaches the photoreceptor, which are the rods and cones. But the photoreceptors sit the furthest back in the retina. So the light essentially goes all the way back and then it moves back forward. Photoreceptors induce the light into an electrical signal via the phototransduction cascade, which we'll talk about in a few lessons. And then once the light is a signal, the signal flows to the bipolar cells, then to the ganglion cells, and then to the optic nerve towards the brain. So photoreceptors are rods and cones. They detect light and initiate the phototransduction cascade. They're at the back of the retina. Horizontal cells, they integrate cells between photoreceptors, allowing for contrast and edge detection. Bipolar cells receive input from the photoreceptors and relay that signal forward. Amacrine cells modulate signals between bipolar and ganglion cells, and they are important for motion detection and temporal sensitivity. And ganglion cells receive input from bipolar cells and send their axons through the retina to form the optic nerve. So essentially, light goes to photoreceptors, which goes to bipolar cells, which goes to ganglion cells. Then horizontal cells are in between photoreceptors and bipolar cells, and they help modulate that reaction. And amacrine cells are between bipolar cells and ganglion cells, and they help modulate that reaction. And these cells work together to pre-process visual information before it even leaves the eye. If you make this into a cartoon, it might better serve your memory. So what I do is I think about polar bears standing on top of fishing rods and those big orange cones. This signifies bipolar cells connected to rods and cones. Then you picture lions standing on top of the bears. So a bunch of lions can be classified as a gang of lions or ganglion cells. This would be ganglion cells on top of bipolar cells. And then you think of the lion's tails all merging and twisting into one, and this would be the axons of ganglion cells becoming the optic nerve. Now where the optic nerve exits is the blind spot in our eye. We don't actively see a blind spot as our brain fills in this image. So feature detection. Our brain doesn't just passively receive images. It breaks them down using specialized neurons tuned to detect specific features. There are three things we consider when looking at an object: color, form, and motion. Form and color are processed by the parvocellular pathway. This has high spatial resolution, but poor temporal resolution. Spatial resolution is detail, temporal resolution is motion. So think pink pyramid is parvocellular. Pink pyramid gives you color and form. And imagine this is like a camera that can only take photos. So you'll get really good detail, but you're not getting any motion. Motion is detected by the magnocellular pathway, which has high temporal resolution but low spatial resolution. So think magnocellular is motion. They both start with an M. And this is like a GoPro in black and white. You don't get great details or resolution, but you get video. And we're going to assume it's good video. Both the parvocellular and magnocellular pathways are found in the lateral geniculate nucleus. This division of labor allows us to simultaneously track movement and see detailed shapes and color. Now, parallel processing is the brain's ability to process multiple aspects of a visual stimulus at once. Color, shape, motion, and depth, they're all handled simultaneously by different pathways. So imagine you combine your camera that cannot take videos and your GoPro. You get an iPhone that can record videos and in good quality. So this is parallel processing. This is how we can recognize a fast-moving, multicolored object without consciously analyzing each component separately. That's it for today. In the next episode, we'll talk about the visual pathway and mapping and color theories.